Dear students, hope you all are fine. We were doing the process of spermatogenesis and we have already covered the process of formation of spermatid. Second part is conversion of spermatid into sperm, or you can call it as formation of spermatozoa. This process is also known as sperm teleosis or spermiogenesis. So if you are asked these two terms in exam, then you should write this is the process of conversion of spermatid into sperm. So here I have shown you diagrammatically, this is the spermatid. Spermatid is a simple normal cell which is almost rounded in shape, having all the cell organelles that includes nucleus, centriole, mitochondria, endoplasmic reticulum, all the cell organelles. And this is the outline of a sperm, which is divided into different parts: head, neck, middle piece and tail. Head contains nucleus and acrosome. Middle piece, neck contains two centrioles. Middle piece contains axial filament around which there are mitochondria, and the tail has axial filament covered by cytoplasmic sheath. Now just see, this spermatid has to change into this sperm. So naturally the process will involve changes in different cell organelles, shifting of different cell organelles, and lots of changes in nucleus, in cytoplasm and in the plasma membrane. So now we will study how this process takes place. The first step is, it's a continuous process, but for our convenience we have divided into different steps. First is changes in nucleus. Nucleus here occupies central position. Now it shifts away from the center. It shifts towards the plasma membrane. So it shifts eccentrically. Then it reduces in volume by losing water. So the process will involve condensation of DNA. So water is lost. Histone and non-histone proteins which are present in the nucleus, they first of all change into transition proteins, and transition proteins, they are later replaced by proteamines, because sperm nucleus contains proteamines. So this is the change in composition of proteins. Here DNA is transcriptionally inactive, means it is not transcribing messenger RNA, and there is no synthesis of proteins from that particular messenger RNA. Only the messenger RNA which has been stored in the spermatid, that is used for the formation of proteins. This DNA does not transcribe messenger RNA. Then nucleus acquires a particular shape and it forms the sperm head. So these are the changes in nucleus. Second is changes in Golgi bodies. So Golgi bodies, they surround the nucleus. First of all, they gather in front of the nucleus. So these are Golgi bodies, they gather around the nucleus. Each Golgi body synthesizes proacrosomal granule. Proacrosomal granules, they fuse to form acrosomal granule. So the membranes of these Golgi bodies, they fuse to form acrosomal vesicle. So acrosomal vesicle contains acrosomal granule. This complete structure is now known as acrosome. This acrosome acquires a particular shape. So this is how this acrosome is formed around the nucleus. So these are the changes in the Golgi bodies. Next is changes in centriole. Here you can say, just see, the two centrioles, they lie close to nucleus. Now these two centrioles, they shift behind the nucleus. So here diagrammatically we have shown, this is the nucleus, this is the acrosome which has been formed by Golgi body. Now the two centrioles, they shift behind the nucleus, one behind the other. Anterior one acts as proximal centriole, posterior one acts as distal centriole. Each centriole is made up of 9+2 arrangement of microtubules that you have studied in lower classes. From distal centriole arises the axial filament. So here we have shown a small part of axial filament. So these are the changes in centriole. Now comes changes in mitochondria. In spermatid, mitochondria, they are randomly distributed in the cytoplasm. Now what happens? They arrange themselves in a spiral manner around this axial filament. So this is the spiral manner. Now mitochondria, they increase in size but they decrease in number. It means mitochondria of the spermatid, they fuse with one another. As they fuse with one another, naturally their number decreases and their size increases. So number decreases, size increases, and they get spirally arranged around the axial filament. So this is the change that occurs in mitochondria. Then what is the change that occurs in cytoplasm? In the spermatid, you can just see there is a large amount of cytoplasm present in the spermatid, whereas sperm is very light in weight. So a lot of cytoplasm is lost, and a thin layer of cytoplasm is formed around different parts of the sperm, forming sperm. So great part of cytoplasm is lost, only a thin layer of cytoplasm is laid down around different parts of the forming sperm. Then the plasma membrane, last change is, what is the change that occurs in plasma membrane? This plasma membrane elongates, and now it covers all the parts of the sperm. So just see, with all these changes, a rounded spermatid having randomly placed cell organelles, it changes into a particular shaped sperm having organelles present in a particular manner. That is, head sperm is divided into 4 parts. Anterior part is head, which comprises nucleus. So naturally this nucleus contains condensed DNA, it contains proteins, and DNA is transcriptionally here as inactive. Then this is the acrosome, which is secreted by Golgi bodies. Then 2 centrioles lying one behind the other form the neck part. From the distal centriole arises the axial filament. In the middle piece there are present mitochondria, which are few in number, but they are large in size, and they are spirally arranged around the axial filament. Then around the different parts of sperm there is a thin sheath of cytoplasm covered by plasma membrane. So with all these changes, spermatid changes into sperm. So this is how spermatogenesis takes place.